Welcome to the deep dive. Today, we're really getting into something quite groundbreaking. It's about how female brains might process past mistakes differently. And it turns out a specific type of RNA, one that's been kind of overlooked, seems to be key. This discovery gives us some genuinely new ways to think about, well, sex differences and depression risk, how we make decisions, even our resilience. Yeah, so our source for this deep dive is a fresh study just out in Science Advances. It's from researchers at Mount Sinai's Icahn School of Medicine. They used animal models to really explore these biological and, psychological links. So our mission today, to unpack this RNA, what it is, what it does in the brain, and really importantly, what it all means for mood, choices, and maybe even new treatments, particularly thinking about women's health. Okay, let's get into it. So let's start with something we know but maybe don't fully understand. Women are diagnosed with depression about twice as often as men. That's a huge disparity. And it's not just how many, it's also how it manifests. Symptoms can differ, right? Like maybe more negative rumination in women.
Exactly. That tendency to sort of dwell on negative thoughts or past events.
Yeah.
And there's been this major gap in understanding, as the researchers put it, about why. Why the difference in rates? Why the difference in symptoms like rumination?
It's a fundamental question.
It really is. And that's what spurred on Dr. Brian Swayze and the team. They wanted to investigate how the brain, specifically a brain potentially at risk for depression, processes prior choices. What's going on differently?
And the way they tackled it is, I think, really interesting. They focused on this group of molecules, non-coding RNA, which honestly haven't always gotten the spotlight.
Right. We usually think of RNA as the messenger, carrying instructions to build proteins.
Yeah, the blueprint.
But these non-coding RNAs, they don't do that. They have other jobs. Think of them more like regulators or switches. They can directly influence how genes are turned on or off or how brain cells function. And they seem to play these really powerful roles in, well, the biology of depression.
So not just passive messengers, but active players.
Definitely. And the specific one here is called LINC00473. It's a long intergenic non-coding RNA. Bit of a mouthful, I know.
LINC00473.
Yeah. And what's really key is what the same team found back in 2020. Dr. Orna Isler was leading that part. They looked at brain tissue from people who had passed away. And in women diagnosed with major depression, they found significantly lower levels of this LINC00473.
Lower levels. Where exactly?
In the prefrontal cortex. That's the brain's sort of executive control area.
Okay. And the crucial part.
The crucial part was this reduction wasn't seen in men with depression. It seemed specific to women.
Wow. Okay, that is specific. So
Yeah.
naturally, the next step was what? See if this holds up experimentally.
Exactly. That's what Dr. Isler did for this current study. She took that human finding and built an animal model around it. And here's a really striking part. When they experimentally increased the levels of LINC00473 in the prefrontal cortex of female mice,
What happened?
it actually promoted resilience to stress, made them cope better.
But only in the female mice.
Only in the females. They didn't see the same protective effect in males. It points to a very specific, sex-dependent mechanism involving this RNA and stress response.
Okay, let's talk about that brain region, the prefrontal cortex. It's the hub for planning, decision-making, linking emotions to actions, basically higher-level thinking.
Absolutely. It's critical.
So if you tweak the levels of this RNA there, how does that actually change behavior? What did they see in the mice?
Well, given its role, you'd expect something to change in decision-making, right? And that's what they found. Altering LINC00473 levels did have measurable effects on behavior. Again, specifically in the female mice.
How so?
They found that the females with higher LINC00473 levels were more flexible. They were quicker to change their decisions when searching for rewards if a previous choice wasn't panning out.
More flexible. Okay. How did the study describe that? They used some specific terms, didn't they?
They did. They used terms from neuroeconomics. They found this molecular change boosting LINC00473 made the female mice more sensitive to what we call sunk costs.
Ah, sunk costs. Like sticking with a bad movie because you already paid for the ticket.
Exactly like that. Overvaluing past investments. The mice with more LINC00473 seem less likely to get stuck because of past effort. And it also increased their sensitivity to regret, a sort of awareness of missed opportunities or better choices they could have made.
Okay, hold on. More sensitive to sunk costs and regret? That sounds kind of negative.
It does sound counterintuitive, doesn't it? That's what's so fascinating here.
Yeah, you'd think more regret equals feeling worse, maybe less resilient.
But the study suggests the opposite. at least in this context.
Uh-huh.
These very traits, being more attuned to past outcomes, both good and bad, being willing to cut losses, might actually fuel greater resilience when facing stress.
That really flips the script.
It does. And importantly, this is the first time we've seen evidence that a non-coding RNA can directly influence these complex cognitive functions. It's not just basic cell stuff. It's shaping decision-making.
Wow. And there was another layer to it, right?
Yeah.
Something about separating past reflection from future planning.
Yes, exactly. Dr. Romain Durand-Cutoli pointed this out. They could selectively change the behavior related to reevaluating a recent choice
Okay.
without actually changing the mouse's initial judgment or preference.
I see. So the process of looking back is distinct from the process of looking forward.
Precisely.
Yeah.
As he put it, choices that depend on reflecting on the past are neurobiologically distinguishable from those that involve planning for the future. And crucially, these distinct processes seem to be handled differently in male and female brains, which then ties back to how emotions might be experienced differently too.
So let's zoom out. What does all this mean? What are the big picture implications here for treatment, for understanding ourselves?
Well, the implications are pretty significant, I think. First, from a medical perspective, this Lin-INC-0473 molecule looks like a potential new target.
For drug development.
Yes, possibly for drugs aimed at depression, maybe specifically those symptoms around negative rumination in women. And it also suggests a potential anatomical target, their prefrontal cortex, for things like brain stimulation therapies, again, potentially tailored for women.
Beyond just treatments, what about understanding resilience itself?
Right. It gives us a potential mechanism, a
Yeah.
biological link between how we respond to stress, our emotional states, and the choices we make based on past experiences. It helps explain why some individuals might be more vulnerable or resilient.
Which brings us back to that intriguing idea from Dr. Swice that maybe sensitivity to regret isn't always bad. How does that work? Because usually we think of rumination, dwelling on the past, as just unhelpful, especially in depression.
It's a really important distinction to make. Dr. Swice suggests that reflecting on the past, even if it feels unpleasant, that pang of regret can actually be adaptive.
Adaptive how?
It can help us learn from mistakes, process emotions, figure out how to deal with stress better next time, grow from experience.
So regret as a teacher, almost.
Kind of. The big challenge now, though, is figuring out the line. When does that reflection tip over from being useful, adaptive processing into unhealthy, maladaptive rumination that actually makes depression worse?
That sounds like a fine line to walk.
It probably is. And that's where this kind of research is so valuable. Findings from animal models like this can push us to ask better questions about human psychology. It could directly inform how psychotherapy is developed or tailored, make it more precise for the individual. And the team is actively doing that, trying to translate these findings back and forth between the lab work and clinical practice.
It really underlines the need for different fields to work together, doesn't it? Psychiatry, psychology, neuroscience.
Absolutely. Dr. Eric Nestler made that point strongly. He emphasized how crucial this kind of interdisciplinary research is. And also the importance of translational research. Using animal models, yes, but always with the goal of bridging back to human health. That's how you accelerate discoveries, get better diagnostics, find innovative treatments.
So to wrap things up then, this deep dive into LINC-00473, this tiny molecule, has shown us a really fascinating mechanism, one that seems specific to females, influencing how the brain processes past mistakes, feels regret, and builds resilience.
Yeah, it's a great example of how understanding these sort of granular biological differences is just so essential if we want to develop more effective, more precise treatments for mental health. We need to move beyond one size fits all.
It really makes you think, doesn't it? Knowing that our biology might shape how we reflect on the past in such distinct ways, how might that change how you view your own self-reflection? Or maybe how you approach understanding why others cope the way they do?
And a whole new layer.
Maybe there really are different flavors of regret, as the research suggests, and perhaps some of them aren't just okay, but actually useful tools for navigating life. Something to consider.